and thank you for joining us. I am Matt Pillar, Chief Editor at Bioprocess Online and host of the Business of Biotech podcast, and I'm thrilled that you've made time to join us. I'd also like to thank Roche Custom Biotech for supporting today's discussion and making it available to you. Today's talk is on risk reduction in mRNA therapeutic development, a topic our guests know very well. Joining us all the way from Germany today are Dr. Christian Dohmen, Senior Director of Formulation and Aerosol Research at Ethris, and Patrick Baumhof, SVP of Technology at CureVac. At Ethris, Christian works on the company's mRNA-based therapeutic candidates for treatment of respiratory viral infections and rare pulmonary diseases, as well as mucosal, multivalent, and mutation-agnostic prophylactic vaccines. He's particularly adept at lipid nanoparticles, R&D, CMC for mRNA ATMPs, and applications for aerosol technology and biopharma, which is a good thing because we got some questions about aerosol technology in our registration process. So, Christian, we'll be turning to you for those. At CureVac, Patrick contributes to the development of drug delivery technologies, controlled release, surface modification of plastics, techniques for purification of nucleic acids, stabilization of mRNA, and even patent law. The company is well known for its incredibly deep development pipeline that comprises 17 mRNA prophylactic vaccines, cancer immunotherapies, and molecular therapies. So during this discussion, we're going to tackle several of the rate-limiting factors that introduce risk to timelines, from chromatography scalability to safety profiles, and from the stability of carrier systems to supply chain challenges and more. And I'd like to remind our audience that in addition to my questions, in fact, more important than my questions, I'm encouraging you to ask questions of Patrick and Christian while they're available to you as a resource. So use that chat box and ask us questions as we go. Christian and Patrick, thank you so much for joining us. You're very appreciated, and welcome.
Thanks, Matt. It's also great to be here, and thanks for the invitation. Great to meet all of you today.
Also happy to be on this panel and looking forward to an interesting discussion. So thanks for having me.
It is our pleasure. We're honored. So I want to start at a pretty high level, and then we're going to drill down into some of the specifics that I just noted. But I want to sort of as a level set, mRNA therapeutics and vaccine development and manufacturing bring with them some formidable challenges that you guys live and breathe every day. We'll discuss those challenges in detail a bit later. But why, in spite of those challenges, is there such high demand for mRNA-based therapeutic development? When I think about it in the context of everything we cover at Bioprocess Online, from ADCs to monoclonal antibodies and vaccine, traditional vaccines and beyond, mRNA is getting a lot of the energy, a lot of the money, and a lot of the buzz right now. So despite these challenges, why so much effort? Where does the promise lie?
Yeah, maybe I can take that question first. Happy to answer. I'm definitely no expert in ADCs or antibodies, so I'm going to speak for the mRNA field. And I think there's simply a high demand for new drugs, especially for until now undruggable targets. And again, just the opportunity to improve the situation for patients. And especially mRNA gives you several advantages that are unmet until now, I think. So there's, of course, the opportunity with mRNA to drug diseases where, for example, protein therapies might not be accessible. For example, intracellular proteins associated to the lipid membrane of a cell. Here, mRNA bears the advantage that the cell produces its protein itself and can guide it to the right direction. And of course, the pandemic and the development of the mRNA field in that area has proven that mRNA drugs can be developed quite quick at a very high quality level and deliver very high qualitative drugs. But I let Patrick speak on that, because on the vaccines I think he at CureVac is one of the experts in that area.
Yeah, I think messenger RNA is a very versatile technology. It can principally encode every protein that you can think of. And compared to recombinant protein technology that is usually dependent on secreted proteins that you can inject, we can also principally build transmembrane proteins. We can also supplement intracellular proteins at more ease, that were usually not addressable with recombinant protein technologies. On the other hand, messenger RNA has a unique pharmacokinetic. If you compare it to, for example, other genetic concepts like viruses or DNA, you have a very defined pharmacokinetic of the expression. It's transient and also has a steep peak and then also a steep disappearance. And in a way, this is also how your body talks. If you have cytokine release or if you have hormones that are usually transcribed by a messenger RNA. And with this unique pharmacokinetic, you can drive biological functions and use them for therapeutic purposes. So we are not limited to vaccine purposes, but we can also manipulate or influence disease states with messenger RNA on levels that are usually not reached by other drugs. For example, we could express transcription factors or other stuff that really can make a difference.
Thank you. So I want to dig right into some of the challenges so we can start talking about solutions and attempted solutions at those challenges. One of the rate limiting factors that I think I've discussed with both of you prior to this call, and I'm interested in your takes on other rate limiting factors, but chromatography came up in conversation, current chromatography scalability being a limitation. So let's talk a little bit about that. Like what are the chromatography limitations that either of you are challenged with right now? Patrick, since you're on my screen, I'll start with you.
Yeah, chromatography principally is a more complex process. You have to apply pressure, and also at some point scaling of the dimensions of your column is a problem. And it's also time intensive and also has some disadvantages within regards of yield in the end. So process time and yield is, especially if you go to the million billion doses or a very large amounts, can be a problem. So, but you can see the current vaccines on the market are delivering in the multi-billion dose range. So it's not prohibitive, but clearly from a standpoint of process development, you would try to find alternatives to speed up and make your production more versatile.